this video is going to wrap up our conversation on the central dogma of biology. So it's going to be a quick recap of what we've talked about, but then I include a slide that has some practice for you because you are going to need to be able to know how to do this on the exam, but it's also going to try to illustrate the relationship between the codons found on messenger RNA and the anticodons found on transfer RNA, and how that helps dictate when and where amino acids get delivered to the ribosome. So let's start with our summary of the central dogma. Remember that the central dogma of biology is also known as gene expression. And so that's because a gene is a piece of DNA. So one long DNA molecule actually has lots of different genes on it, okay? So just think of one big piece of DNA, and you could say, oh, the first part, here's gene 1, then there's gene 2, then there's gene 3, so on and so forth. And so remember, each gene codes for its own protein, okay? And so the purpose of gene expression is to express the protein that the DNA gene codes for. Okay, and so in order to do that, we have to look at the piece of DNA. Okay, and for simplicity, they divide the DNA, as you see in this picture, the DNA nucleotides into 3s, and that's only because when we eventually get to the RNA part of this conversation, right, we group them in 3s. So it's just for simplicity that they're doing that in the DNA picture as well, okay? So we're gonna express the protein coded for by the DNA, the gene. So remember, the first step in doing that is to use the template strand of DNA, which is the strand running 3' to 5', and using that as a template to create a piece of RNA. In this case, we're worried about messenger RNA, and the messenger RNA is always made 5' to 3', okay? And so the process of using a template strand of DNA to create the messenger RNA is called transcription. So once we have the RNA sequence, we can kind of like draw it in sets of 3. And remember, those sets of 3 are called codons. And those codons tell us which amino acid we need. And to know that, we would use the codon wheel that will be provided. And so using that, we can go through the process of translation, which again is where the transfer RNA will bring the correct amino acid to the correct place in the ribosome. So remember, for transcription, which is when we're making RNA, there's only one key player, RNA polymerase. RNA polymerase is going to read that template strand of DNA and add a complementary, and build the strand of RNA using complementary nucleotides. So if there's a guanine on the DNA, a piece of RNA is going to have cytosine. All right, so then again, the key players for translation are going to be that messenger RNA that was just made during transcription, and then tRNAs and the ribosome. And at the end of translation, we have a polypeptide chain, which is just a chain of amino acids that will eventually fold into a specific 3D shape. At that point, technically, we can call it a protein. Okay, so that's just big picture what we've been talking about. Let's go ahead and have you practice going through this process on your own. So for right now, ignore all the little things at the bottom right, okay? So there's your template strand of DNA. I did not include directionality on it because I want you to tell me what the directionality has to be. So at this point, I suggest you push pause, you write out the template DNA sequence, add in the directionality, and then fill in what messenger RNA would be made as a result of transcription. And then after that, what would be the amino acid sequence of that polypeptide chain that would be made during translation. Now, I'm going to go ahead and continue with this video, but I do suggest you pause it and do it on your own. It's as much easier to do when I'm doing it for you, but it's really important that you know how to do this without hesitation for the exam. All right, so here we go. So we know that messenger RNA is always made 5' to 3'. So that means our DNA template strand is 3' to 5'. We also know that that template strand is DNA because it has thymines. But remember, RNA does not have thymine, it has uracil. So as we go through and write this messenger RNA sequence, you're not going to have any thymines, okay? Instead, adenine will pair with uracil, okay? So looking at the DNA, we see the first nucleotide is thymine. So we know that the RNA must be complementary, so that's going to be an adenine. Now, the second nucleotide in DNA is adenine. So what does that pair with for RNA? Uracil. And then we have cytosine as our third DNA nucleotide. And what pairs with cytosine? Guanine. Now, you know you're doing this correctly because AUG is always the start codon. So that should always be what you start with your RNA sequence. So I'm going to go ahead and fill out the rest, okay? You can go ahead and pause this and check your sequence. I went ahead and added that 3' directionality as well, because with nucleic acids, there's always directionality. So now what we need to do is basically section off the messenger RNA sequence into sets of 3. Remember, those are called codons. So we have 6 codons in this little piece of RNA. Now I'm going to go ahead and get rid of those. And so now we need to use the codon wheel to figure out what each of those codons code for, right? We're trying to figure out what amino acid do we need if the codon is AUG. Well, hopefully you remember that the start codon, AUG, always gives us the amino acid methionine. So essentially what's going to happen here is if you look at the bottom left, or I'm sorry, the bottom right of this slide, I drew little tRNAs. They normally kind of have this little kind of cross-like shape. And that was the best I could do in PowerPoint. And at the top region is going to be the anticodon. And at the bottom is going to be the amino acid. Now, I didn't show the directionality of the RNA for the tRNA. And they're arranged this way just for simplicity, right? Because I need my anticodon found on the tRNA to be able to line up with my codon on the messenger RNA, okay? So remember, there are 2 sites on the tRNA, and that is going to be the amino acid attachment site. And again, on my illustration, the amino acids are on the bottom of the tRNA. And then at the opposite end, it has the anticodon. And you should be able to recognize that, right? Because those are the RNA nucleotides. So that's the anticodon. So we said that our first codon is AUG. And if you use the codon wheel here, right, you always start in the middle. So there's AUG, and that's methionine. So essentially what's going to happen is any tRNA, or I'm sorry, a tRNA that's carrying methionine is like, ooh, that's me. Here I come. And it's going to bind. And the reason that methionine gets dropped off is because the anticodon of the tRNA, which you see in black, is complementary to the start codon on the messenger RNA. So the only thing that can bind here and deliver an amino acid is the UAC anticodon, and that's always connected to a methionine. Okay, so that's how we ensure that methionine is going to go first. Well, now our second codon on the messenger RNA is CGU. So use the codon wheel. CGU gives arginine. So this signals to the cell that, hey, I need a tRNA carrying arginine. So wherever one is in the cell, it's like, oh, me, here I come, bloop. Okay, and it attaches. Now, this model that I'm doing right now isn't going to be completely perfect. I'm not showing the ribosome. I'm not showing the actual attachment of the two amino acids together and, you know, the tRNAs disappearing after they've done their job of dropping off the amino acid. The purpose of this illustration is to really drive home the connection and relationship between codons and anticodons and the amino acid, okay? So now our third codon on the messenger RNA is AUU. Use the codon wheel. AUU is isoleucine. So now the tRNA, wherever it is, that has isoleucine can come and bind to that codon. Okay, because again, the anticodon is going to be complementary to the codon. So now you tell me, our next codon is CUG. So what should the anticodon be on the tRNA that's going to deliver that amino acid? To answer that, you just have to remember your base pairing. If the codon is CUG, the anticodon is opposite. So GAC. Okay? And so that's what you're going to find. That anticodon is, in fact, GAC, which is opposite of our CUG codon. And if you look, CUG as a codon should have delivered us leucine, and it sure did. Okay? Finally, let's see. Hold on a second. My thing's put all out of whack. Okay, so the next up is GUA, and that is going to give us the amino acid valine. And so the tRNA carrying valine, oops, sorry, that's a little out of whack, but that's okay. It's going to come, and now we've got all of our amino acids delivered so far. Then we get to our final codon, which in this case, it's the last one, so it's going to be the stop codon. And remember, there's no amino acid called stop. So there's no tRNA associated with this stop codon. Instead, that's where the release factor would bind. Everything would release from each other, and the process is over. So for the amino acid sequence, it is strictly methionine, arginine, isoleucine, leucine, and valine. And of course, you don't have to spell those out. You would just put these 3-letter codes that you see. But do not write stop, okay, because there's no amino acid called stop. All right, that wraps up our discussion on gene expression. The next set of videos will talk about like mutations. So you have to understand this DNA, RNA, amino acid sequence, process in order to fully grasp mutations. So make sure that you can do this type of practice problem on your own before watching the mutations video. All right, y'all, let me know if you have any questions.